So what is it that makes you, you? The answer, and it's a wild one, is hidden deep inside your body. It's written in a chemical code that's billions of letters long. Today, we're going to crack that code. We're going to trace the path all the way from that tiny blueprint to the living, breathing organism it creates. I mean, it's a question we've all asked, right? It's the perfect way to start thinking about genetics. That unique mix of traits you got from your parents, well, that's the most personal proof you'll ever get that this code is real and it's at work. And to really get how it all happens, we've got to go right to the source, the blueprint of life itself.
Okay, let's get into it. We're talking about the ultimate instruction manual. Seriously, it has every single piece of information needed to build and run a living thing. Doesn't matter if it's a tiny bacterium or a massive blue whale.
And there it is, DNA. You've all seen this iconic shape, right? This beautiful, elegant double helix. That is the molecule carrying our genetic code. A good way to think about it is like a gigantic library. I'm talking billions of letters, and every single sentence, every paragraph, holds the precise instructions for a specific part of you.
So if DNA is the whole library, then what's a gene? Well, a gene is like a single recipe from one of those books. It's a very specific chunk of that DNA that codes for one particular thing. You know, like the recipe for making the brown pigment in your eyes, or the instructions for building a protein that helps you digest your lunch.
So this language, it's surprisingly simple. It's built from just 4 chemical letters, or bases, that make up the rungs of that twisted ladder: A, T, C, and G. But here's the really cool part, the part that makes it all work, the pairing rule. A always pairs with T, and C always pairs with G. No exceptions. This super strict, totally predictable chemical partnership, that is the secret to life stability. It's what ensures the code can be copied perfectly, millions and millions of times, with almost no mistakes.
Okay, so we have the blueprint, but how does a cell actually read it and turn all those instructions into, well, you? The answer is a process so fundamental to life, it has a pretty serious name: the central dogma of molecular biology.
And it's this really brilliant 2-step system. First up is transcription. The cell makes a temporary copy of just one gene. This copy is called messenger RNA, or mRNA. You can think of it like this. The DNA is a priceless, ancient book that can't leave the library vault, so you just photocopy the one page you need. That's mRNA. Then, step 2, translation. That photocopy, the mRNA, travels out to the cell's factory, the ribosome, which reads the instructions and builds the exact protein that the gene was describing.
Francis Crick, one of the guys who discovered DNA's structure, summed the whole thing up beautifully. DNA leads to RNA, which leads to protein. That's it. The permanent DNA archive gets transcribed into a temporary RNA message, and that message gets translated into a protein that actually does something. And trust me, these proteins do pretty much everything in our cells, from building our muscles and bones to fighting off germs and carrying oxygen in our blood.
So what's the real difference between the master blueprint and that working copy? Let's break it down. DNA, right? It's that stable double helix. It's built to last. Think long-term storage. RNA, on the other hand, is usually a single strand. It's more disposable. Perfect for a quick, short-term message. There's also a tiny chemical difference. RNA uses a letter called uracil, or U instead of thymine. But the big takeaway here is this. DNA is the protected master file, and RNA is the quick, expendable memo you send out to get the job done.
All right, so we've got a handle on how the code works inside one single organism. But what about the bigger picture? How does that blueprint get passed down from a parent to their kid? Well, that's where a very, very special kind of cell division comes in.
And that process is called meiosis. Its whole job, its entire purpose, is to create reproductive cells, sperm and eggs. Here's a way to think about it. Most of your body cells have 2 full sets of your genetic encyclopedias, right? One from your mom, one from your dad. That's called being diploid. Meiosis is the process of making cells that only have one complete set, that's haploid, so they're ready to pass on. That way, when a sperm and egg combine, the new baby has exactly the right amount of genetic info. Not too much, not too little.
And this is where the real magic starts to happen. In the first big step of meiosis, the cell lines up its chromosome pairs. Remember, one from each parent. But instead of just copying everything, it does something way more clever. It actually separates the pairs, making sure that each new sperm or egg cell gets a complete but totally unique mix of chromosomes from mom and dad.
See, meiosis isn't just about cutting the chromosome number in half. It is a powerful engine for creating genetic diversity. Two incredible things happen. First, something called crossing over. The paired-up chromosomes literally hug each other and swap pieces of DNA. They're shuffling the genetic deck, creating brand new combinations of genes. Then, there's independent assortment. When those pairs line up to be separated, they do it totally randomly. This alone creates millions of possible combinations. And this is the big so what moment. This is the chemical reason why you and your siblings aren't identical copies of each other unless you're identical twins, of course. Okay, so we've seen how all this gene shuffling happens on a tiny chemical level. But how does that, translate into the stuff we can actually see, like eye color or height? To answer that, we have to take a little trip back in time, to the garden of a 19th century monk.
Meet Gregor Mendel. He was a monk who had a serious passion for pea plants. Yeah, pea plants. Now, this was long before anyone had a clue about DNA, but Mendel, just by studying his peas, figured out the fundamental rules of heredity. And what he discovered in that quiet monastery garden, well, it would change science forever.
So here's what he did. He took some pea plants that only ever made purple flowers and crossed them with plants that only ever made white flowers. Simple enough. But what happened was weird. All of the new plants had purple flowers. It was like the white flower trait had just vanished. But, and this is the kicker, when he let those new purple plants pollinate themselves, the white flowers came roaring back in the next generation. And not just randomly, but in a super predictable 3-to-1 ratio.
Now, to explain this, Mendel came up with an amazing idea. He proposed that for any given trait, there were different versions of the factor that controlled it. Today, we call those versions alleles. Think of them like different flavors of the same gene. In his peas, the purple flower allele was dominant, meaning if a plant had even one copy, its flowers would be purple. The white flower allele was recessive. It was still there, but it was hidden unless a plant inherited 2 copies of it. And just like that, he explained how a trait could seem to skip a generation.
And here it is, folks. This is the key connection we've been building towards. That microscopic chemical process of meiosis, the shuffling and separating of alleles into sperm and eggs, that is the engine that directly causes the big predictable patterns that Mendel saw in his garden. That 3-to-1 ratio he found, it's not magic. It's the beautiful mathematical result of chemistry doing its thing. For hundreds of years, all we could do was observe these patterns, maybe predict them, just like Mendel did. But in just the last few decades, everything has changed. A revolution has happened. We've gone from being just readers of the genetic code to being its authors.
I mean, think about that. That is a monumental shift. For the first time in the billions of years of life on this planet, one species, us, has developed the ability to not just read the book of life, but to actually pick up a pen and start rewriting it.
And the possibilities are, well, they're staggering. What if your doctor could prescribe medicine tailored to your specific genetic makeup? What if we could actually fix genetic diseases at their source before they even start? What if we could design crops that can survive a drought and help feed a growing world? These aren't sci-fi questions anymore. This is the reality we live in now, in the age of biotechnology. So, we now have the tools. We can literally rewrite the blueprint of life. But with that incredible, almost unbelievable power comes a profound responsibility. It leaves us with some pretty big questions, right? What future are we going to choose to build? What stories will we decide to write in that book of life? And maybe just as important, who gets to decide? The future is really in our hands now. So, what are we going to do with it?